In the title of the talk, I mentioned learning structure and mouse behavior. Very simply speaking, the brain governs our behavior, and as neuroscientists, we're interested in the biological mechanisms that make that possible. In this example, a mouse is traversing its natural environment. It's relying on cues from the periphery to navigate and behave appropriately. In order for this animal to accomplish this, it relies on the appropriate interaction of genes and neural circuits, which, due to relatively new advances in neuroscience and genetics, we are able to measure and manipulate quite well in the laboratory. However, it's traditionally been much more difficult to quantify behavior. Originally, ethologists observed behavior in the wild, much like we're doing now, looking at this video of the mouse looking for a nut, and then pushing a leaf over, and then maybe grooming a little bit. And they would write a few sentences about what that animal was doing. And they would spend hours and hours at a time doing this. So given these observations and careful kind of annotation of what was going on with this behavior, famous ethologists such as Nico Tinbergen and Konrad Lorenz intuited that behavior is actually modular. It's made up of parts, and hinted that this could be actually a better way to investigate the behavior of an animal rather than writing a few sentences describing globally what's going on. So to that end, Kent Barrage's group and many others have been interested in investigating the modularity of a very specific behavior, which is grooming. So here is a plot at the top showing paw trajectories during a grooming sequence. So the small circles there you see are these kind of symmetrical small circles where the mouse is rubbing its nose, and those longer symmetric-ish trajectories, which are these guys here, is the mouse reaching back behind its ears and then reaching all the way to the front. And underneath, you can see hand-annotated labels by experimenters indicating that these blocks of action are actually reused over time. So this sequence goes on and on and on. I want to highlight here that this is great work to show the modularity of behavior, although this was not easily done. So this took some painstaking work. This is high-speed videography, where experimenters went through frame by frame and annotated exactly what the mouse was doing at any given point in time, which is a major limitation to doing this in our laboratory and others. So this body of work, this grooming body of work, has continued, and now there's a decent-sized literature on the neural mechanisms involved with many aspects of the grooming behavior, such as brain areas involved in sequencing, like the striatum, which Jeff will tell you a little bit more about later, and pattern generation, like the cerebellum. In a recent review, authors summarized the translational importance of this work. So since we're able to make genetic mouse models for a variety of diseases, illustrated in this plot here as blue dots, it's now possible to see specifically how grooming behavior is modulated from a typical state, which is at the center here, and what areas we might need to investigate further and eventually target with therapies to fix that behavior. So for example, on this plot, basal ganglia disorders, which are down here, which are largely movement disorders, appear to occur at the same activity level as a typical animal located in the center. However, the patterning of the grooming sequence itself is much more flexible, meaning that it's a lot more variable. So this would be a hint to us as experimenters to perhaps investigate and target areas of the brain involved with sequencing, like the striatum, which many groups are currently doing. So, oops, nope. This video isn't working. There we go. So the work I showed you, I think, shows why studying the modularity of behavior is important. However, that work only focuses on a small portion of the mouse's actual behavioral repertoire. So looking back at our mouse friend here, in navigating in the wild, we can appreciate that behavior is quite complex and highly dimensional. So in the laboratory, people study many different aspects of this behavioral complexity. So in this case, we are investigating, well, not we, another group is investigating the willingness to swim as a kind of a measure of depression in a mouse. In this example here, this is called a rotarod, and this is measuring endurance and motor coordination. You can see actually mouse 3 here is going to do a backflip really soon. There we go. And probably the most commonly used test in the lab is the open field test, which is supposed to represent a kind of naturalistic environment. If you look at its behavior now, it's doing similar things to what the animal is doing out in the wild, although there's no leaves or nuts or berries around. And usually what happens is, someone puts a mouse in this bucket and is interested in a feature of its behavior, such as, say, where it is in space. You can draw a little trajectory of where that mouse moves around and then quantify some feature of that trajectory. For example, how much time it spends in the middle versus how much time it spends on the outside of the arena, which is supposed to be kind of a proxy for how willing the animal is to explore the center and another measure of kind of anxiety, depression.
If you did a random walk, would it give you the same picture?
So the question was, if I did a random walk, would I get the same picture? As far as the mouse trajectory plot goes here,
like the graph.
The graph itself, I'm not sure. Yeah.
So when you first drop a mouse in an open field, it doesn't typically like to be in the center. Because the center is this big open thing, right? Where any bird could swoop down and hypothetically capture the mouse and take it away and eat it. So there is, there's some non-uniformity in where it wants to be when you just drop it in a naive mouse into the open, right? But eventually it'll get habituated, and maybe you can better model it with something like a random walk.
So your comment about that would, I might be taking this aside, but I would imagine it would be more scary to be stuck at the edge, because then it has less ways to escape a predator. So that would counter your thought.
Sure, right? That's like your intuition, maybe if you were in a bucket, but I think mice like to hide, and usually the predator has to worry about the most are predators from above, right? So there actually are well-known responses to things that are looming from above it. And these are things that'll just snatch a mouse, without it seeing it coming, right? Whereas if there's something like a fox or a dog or a cat or whatever, maybe it might be right in front, and at least it can see it. So yeah, of course, like none of these rules are perfect, right? It's a mouse in a featureless bucket, right? So maybe it'll interpret things ethologically the first 10 or 15 minutes it's in that bucket, but eventually they habituate to it, and they're like, oh, I'm in something where I'm not going to be eaten, especially the 4th or 5th time they're dropped in there. Right? And then they just might, walk around somewhat randomly. But at least with some stickiness to where they're walking. It's not as if they walk 2 steps. Like, they're not like E. coli, right? Where they're moving in one direction and they randomly switch that direction and then take another 2 steps, right? So.
Good. So we have a mouse trajectory, and we quantify some feature of that trajectory, like the amount of time it spends inside versus outside. And then we can compare across mice and do a t-test and see that there's some significant difference between the amount of time that the mouse is spending inside versus outside. So there's a couple issues to this. One major issue is that we quantify these low-dimensional behavioral features mostly painstakingly by eye, like the original example I showed you of grooming, and make a conclusion about how an animal's behavior is changed as a consequence of some experimental manipulation, like a drug or a stimulus. However, these metrics don't really represent the mouse's behavior at large, and again, are really low-dimensional representations of the bigger picture. So to that end, our lab is interested in developing a platform to kind of address all of these limitations. So we developed MoSeq, motion sequencing, to quantify high-dimensional 3D data, 3D mouse behavior data, objectively and with granularity. And at a very high level, the MoSeq framework kind of looks like this. So you record a mouse moving around with a depth sensor, you do some time series modeling, and you get out this very granular picture of behavior. I'll go into more detail, of course. So that will be kind of the agenda of the talk today. So now I'll take you through the pipeline, which involves, broadly speaking, these 4 steps. Now, usually when explaining this technique to biologists, they don't care too much about steps 2 and 3 here. They just kind of want to know that there is some sort of button that you're pushing to get out some output that you're interested in. So today I will try to unveil what's underneath the MoSeq button for you guys, and we can have a discussion about those features. Okay, so on to 3D imaging. As I showed you, how do we get the data? So as I mentioned, we use depth imaging to record mice. So we use a Kinect sensor, which you can just buy at Best Buy. Actually, this sensor isn't available at Best Buy anymore, but any depth sensor will work. So we've used Intel RealSenses, Kinect version 1, other stereo cameras, whatever will get you a depth image. And just very briefly, oops, yeah, there we go. There's a mouse navigating a bucket. And just so very briefly, time of flight works by pulsing in IR light and measuring the amount of time it takes for that IR light to travel back to a sensor. And then from there, you get this nice depth image here that's 512 by 424 pixels with a bunch of 16-bit integers that represent the distance from the sensor here. Okay, so why do we use the bucket? So data is recorded in the bucket because the mouse displays a rich variety of behaviors similar to the video of the mouse in the wild from earlier. It's also a pretty friendly imaging environment, so we can get clear depth information with limited noise. And we could also use more complex arenas too. We can't yet use more complex arenas like with naturalistic stuff in there, like leaves and nuts and whatnot, because it represents a pretty challenging image processing issue. We're working on it in the lab now, but for now, the simplicity of the bucket is good for us.
Have you ever tried different shapes of bucket?
Yeah. So we have circular arenas, square arenas, rectangular arenas, some with different features. I'll show you an example later of an arena that has odor being presented to one corner of the arena. So you can see other behavior changes as a function of that odor. And this cage system you see on the right-hand side is equipped to hold equipment to measure neural activity that Jeff will talk about in a little bit more detail later. So we can get simultaneous behavior and neural activity recordings, which is what our custom software here does. So it uses a National Instrument data acquisition board. So you can use all different types of sensors to align in time that sensor information with the 3D mouse behavior itself. And so at the end of the session, say you record the animal for 30 minutes or so, you get a folder with some raw depth data and some timestamps, metadata, and other NIDAC information like neural data or anything else that you're recording. Okay, so that's kind of the brief overview of how we acquire the data. Now we'll get into what we're actually doing with the raw data after we get it. So as I showed you before, we have this image here, this movie here of the mouse navigating the bucket. This is pretty highly dimensional, so first what we want to do is just crop the mouse out of this image here and orient it so it's always facing towards the right-hand side. And this data is saved off to an 80 by 80 by n matrix where n is just the number of frames that you're recording. In parallel to that, we're also extracting some movement features from the mouse itself as well. So features that I mentioned earlier that were interesting in that open field experiment. So like velocity, the 2D velocity, 3D velocity of the mouse, the height, the length, the width, and the positional information. So where its body is in the arena and like an XY coordinate, for example. And we still do some of that kind of low dimensional analysis. For example, this is a histogram of the mouse's height over the course of the experiment. You can see that the orange guy here spends most of its time in one of two kind of height states, I guess maybe you would want to call them. Whereas this blue guy here is a little bit more trimodal, so maybe it's spending more time rearing up, and that would be a good kind of proxy for that. Also, we can make just general heat maps of the data to show where the mouse is moving around. Again, this isn't really a robust picture of what the mouse is doing, like modulating its pose-wise. But it is kind of a useful quality check to be sure that your mouse is moving around and exploring the arena. Sorry. Okay, great. So even still, dimensionality of this matrix is still pretty large. So what we want to do is reduce the dimensionality. So to do that, we transform this matrix into a 6400 by n shape matrix. We compute PCA on this matrix using probabilistic PCA. And then we get a lower dimensional representation of the mouse images themselves in this array here. And because this is still pretty highly dimensional, we use some computing resources that Jeff and others in the lab have worked out, such as Dask, that works well on a Slurm cluster to parallelize a lot of the PCA computation. So then, taken together, what I've described to you so far is that you have some raw data of a mouse moving around in a bucket. We crop and orient that mouse so that you get a big matrix of the mouse stationary, but modulating its pose over time. These are simply compressed raw frames unraveled to show that there's some blocky structure to the data. And then here's an example of the principal components that we computed overlaid to those raw frames. And what you might be able to appreciate here is that there's some structure here. And there's no real sense, actually, whether or not this structure is reused over time. But I think by squinting, you could tell that there are some kind of unique features here.
Is that about the real dimensionality that you have on the bottom? Is that the typical number of dimensions that you would extract through
Yes.
the PCA? Okay.
Yes, typically we use 10 principal components. 10 principal components represents 90+% of the variance in the data. Okay, and by the way, the PCs is what we're going to be modeling here. So this is the time series that we're interested in. Okay, so moving on to the modeling itself. So can we use a time series model to learn discrete states in the principal components? So to answer that question, we looked at the literature and saw that ARHMMs are used a lot of the time in signal processing, like speech analysis, or in econometrics, like trying to predict, like stock movement. And so an autoregressive model will predict future behavior based off of past behavior, and an HMM will help us approximate latent states given some observable variables like the PCs here. So basically what we do is we feed PCs to the ARHMM, and what we get out are these discrete states here that represent changes in the PCs here that also match up well to the raw data as well. Is that all making sense so far? Okay. There is, I guess, a caveat to the ARHMM. we say this is unsupervised learning, which is kind of true. There's actually a tunable parameter, a tunable free parameter called kappa here, which helps you focus the time scale of the behavior that you want to discover. So what we do is we scan over this kappa parameter to match the duration distribution of the syllable states to the duration distribution of the raw data states here. So this is a duration distribution of the raw data. You can see that on average, states are occurring with a length of about 350 milliseconds. And what we do is, scan over kappa, compare the duration distributions using like Jensen-Shannon divergence, and pick the model that overlaps best. Yeah?
Could that really break down if you had really heterogeneous time scales of behaviors?
Yeah, that's an interesting question. So the question has to do with the time scales and behavior. So that's something I'll touch on a little bit later in the talk. That is an interesting research question in and of itself. We think that these states represent, basically the fundamental units of behavior. I mean, maybe that's a stretch to say. We can investigate as well using cameras with higher frame rates as well. But yeah, we are interested in this hierarchical kind of whether or not there are behaviors occurring at multiple timescales. Yeah.
I mean, just to put a point on that, so kappa's not fixing a single timescale, so these states can all self-transition. So if the mouse falls asleep, it'll just basically self-transition forever. There's obviously a limit to how heterogeneous things can be, and so there are extensions of this type of model where essentially you can add additional layers to the discrete latent states and then capture basically one time scale behavior, a slightly coarser time scale, coarser yet again. So it's fairly straightforward, I think. Not straightforward mathematically, but straightforward conceptually to extend these models to account for explicitly a hierarchy of time scales. And you can account for some heterogeneity just with the AR HMM as it is.
Okay, so taken together, very simplified. What MoSeq does is take 3D information from a mouse, reduces the dimensionality of that mouse moving around in a bucket to some number of principal components. And then we model those principal components using an autoregressive hidden Markov model, which then returns what we call behavioral syllables. So these are just the state labels that the AR-HMM returns. But what those actually represent are discrete behaviors here, which is what we as neuroscientists are really interested in better understanding. Okay, so into this whole behavioral syllables concept.
Jeffrey, sorry, Ralph, are you
Yeah.
going to say a bit more about what a state is in this AR-HMM?
Yeah, that's a good question that I think Jeff is probably best suited to answer.
So do you want to say like a sentence or
Yeah.
two, and then I think you'll probably have more time in your talk. But
Yeah, so the essential components are that you have the hidden Markov model and discrete latent states in the hidden Markov model. So we're all fairly familiar with those. And the thing that's slightly different about this model is that the emissions are autoregressive. So the observables are continuous in time, those are the principal components, and then behind those are discrete latent states that are switching between different autoregressive processes, right? So you can have different types of dynamics that represent different behaviors, and those are the states, these discrete latent states in the HMM. So just as if you typically you might use Gaussian emissions in a hidden Markov model, and you have discrete states and observable states, these are the discrete states. Okay, so when we say state, when we say syllable, these are the discrete latent states of the AR-HMM. Is that point? That was like 5 sentences.
Great. So making sense of these behavioral syllables. So much like the grooming example, we now kind of have a parts list we like to call for for most body language, in a sense. And we can use this information to start understanding and quantifying behavioral structure in many different contexts. So stuff that we're always interested in as behavioral neuroscientists is something very simple, which is just how often is each behavior used. So on the x-axis here, we have syllables that are roughly ranked by usage. And this is a bunch of mice recorded in a bucket. I think the n is 10 or something. And you can see that this represents somewhat of a behavioral fingerprint, so to speak. Usually the first couple of syllables are locomotion-based syllables. And they're usually different flavors of the same syllable represented. So, there could be 5 different syllables or states that are all rears, the most rearing up in the bucket. So if we go back here, yeah. So there could be 5 different states that are rear up. There could be a couple different states that are scrunch, a couple different states that are this jumping behavior that all have their own characteristic flavor. Maybe one is a rear up to a kind of fan on the wall. Maybe another one is run forward to a slight turn at the end. Yeah, and the other thing that we're always very interested in is the transition structure of the behaviors. So usually we compare, and I'll show you in more detail in a couple slides, we can compare the transition statistics between conditions to get a better sense for just like what is the structure of mouse behavior itself. Okay, so now I'll describe the experiment. Oh yeah, question?
Wait, you said that like when the mouse gets dropped in the bucket after a bunch of times, the behavior might be different. So like for these, are you taking data from mice that like it's their first time in the bucket, or like do you use this same amount of experienced mice?
Yeah, so the question is about whether or not we're using the same mouse over and over again. No, so we're using different mice, and they're all naive to the environment. Although this definitely depends on the research question that you're interested in. If you're interested in this kind of chronic bucket behavior over time, or you're doing like a chronic drug study where you want to see the mouse's behavior evolve over multiple days in the bucket, then of course you could test that. As long as you model all of the, if you co-train the model on all of the data together. Okay, so in the Datta Lab, we're largely interested in odor processing in the brain and how sensory information is converted into behavior. So we did a simple experiment where you have a square arena, and in one corner of the arena, you pipe in smell of a predator, smell of a fox, which is this TMT here, trimethylthiazolene. And this compound has been known to induce innate aversion. So typically what we do is, so we can record a bunch of control animals where there's actually not an odor in here, there's a blank odor. And record a bunch of animals where we pipe in the fox odor and then compare their behavioral fingerprints, so the output of the model.
So reiterating her question, do you expect something different, or is this checked where you first have the mouse in the bucket, no odor, it gets used to environment, and then you drop in the odor, versus the odor is there to begin with, and then the mouse starts naive in the bucket with the odor there?
Yeah, so in this case, the mouse is dropped into the bucket when the odor is being piped in. And it's the first time that they will have experienced that environment. So we record 10 animals or n number of animals where that is the case, and then n number of other animals where there's simply no odor going into the corner. Does that make sense?
Have you also considered that third scenario where the mouse is in the bucket for a while and then the odor drops in?
Yeah, so we have done that. We've internally controlled this experiment as well, and the results are quite similar. Yeah. Yep.
Is the idea that you fit one model that represents all of mouse behavior and then see how unusual it is, or are you fitting models to each of these individual scenarios like TMT versus no TMT?
Yeah, so the question is whether we fit one big model or individual models on for example, one condition versus another. So we fit the model on all of the data together because we wanted to learn the global mouse structure. And then from there, we can begin to see which behaviors are up and down regulated in each condition. And so when you do something like that, what you can begin to see is that, I'm not depicting it here. I should have overlaid another set of points here, which is control versus TMT. My apologies. But imagine there is another set of points here, and there are some substantial differences. What you can do then is pull out those differences and create a little movie of what that difference is. So in this case, what I'm showing you is, I guess I didn't describe that in the other movies either. Sorry about that. We call these crowd movies. This is just a single instance of the syllable occurring, overlaid on top of each other to give you a representation of what the behavior actually is. So when the white dot goes on, the mouse is in that syllable, is doing that syllable. So in this case, what you see is there's an upregulation of freezing behaviors in the TMT case. As you might expect, it's getting experience with the predator, and it kind of isolates. It wants to hide in the corner. There's also another behavior which you also might expect, which is odor-guided avoidance. So the source of the odor is here. So what this is representing is the mouse smelling the odor and then darting away. So continuing on the kind of idea that this is, that we're investigating a mouse body language, what you can do is start to really better understand what the structure, what the grammar of that language is by representing each state as a node on this state map, and then showing the interconnectedness. So the thickness of each one of these lines represents the probability that this state is going to transition to this state, and the diameter of these circles here represent the usage of that state overall. So as you might be able to appreciate, it looks like the TMT animals are occupying this transition space that is not really occupied in this control animal. So very simply, we can just take the difference of the two and actually show that there are substantial differences in the transition structure between both of these groups. And just a point on this, this is the type of framework that we use to investigate a bunch of different questions. So this doesn't have to be predator odors. This could be drugs, or this could be a genetic mutant for any number of diseases, or this could be manipulating a specific neural circuit versus not manipulating that circuit. So this is really the reusable part of the pipeline that we continue to use. So that's the basics of the original pipeline. Now, there are a number of ways that we're thinking about extending this pipeline. One kind of interesting avenue of research that is gaining some steam right now is studying social behaviors. So to that end, our lab, new postdoc in the lab, Caleb Weinreb, in collaboration with Scott Lindemann, who I think some of you might know here, are working on modeling social behaviors. So we can automatically segment each mouse, and then we can extract and orient the mouse images here and use the same pipeline to then create, to then train a model and learn mouse syllables in this social context. And what we're interested in here is actually whether or not these two animals start to develop these are two males, so whether or not they start to develop a social hierarchy. So you can see actually in this image here, they're starting to fight a little bit. And this is interesting for a number of reasons from a neurobiological perspective. There are a lot of people that study circuits for aggression and social behavior and whatnot. So what we can do here is train a model on all of this data and show most 1 and most 2 overlaid on top of each other. They're behavioral fingerprints here. The first session that you put them in together. So this is brand new. They haven't met each other before. You drop them in together. And you can see that they're pretty similar. Maybe there's some states here that are quite different. But if you fight them fight them. This is not a mouse fight club. If you record them multiple days in a row, you can appreciate that the difference, the behavioral variability between the two is increasing. So just anecdotally, what we see is that a clear hierarchy is established after the first day, where the submissive mouse will kind of stay frozen like a statue almost, and the dominant male will run around and sniff it and then fight with it. You can see that a little bit here in the bottom left, where this guy is the submissive one, and this guy is the dominant one. So he's kind of chasing him. So anyways, those behavioral differences are reflected in the behavioral syllables themselves. And then briefly, just to wrap up, this touches on your question about potentially multiple timescales. So if we have a bunch of syllable labels here, you might suspect that sequences of behaviors are actually used over and over again, and why don't we just model those behaviors? Like I said, there's multiple different types of rears and grooms, so why not just collapse all those together and call them one state? So we're working on some computational techniques to do that. That's work from Diego and Scott Linderman, where you can see here, this is the state space of the hierarchical model and the original model itself. So you can see that there's a lot more switchiness here. And in the hierarchical model, spending longer time in each one of these states. So it's finding this higher order structure in the data. Okay, and that's all I have for today. Thanks for bearing with me on all of the experimental details. Yeah, I just want to acknowledge Bob Datta, who's been a great mentor. The MoSeq version 1 makers, who I think a lot of you might recognize. I think some of them have given talks here. So Alex Wilshko, Matt Johnson, who are both now at Google Brain, Ryan Adams, who's at Princeton now, I think. And then the MoSeq kind of V2 plus makers. So Jeff has extended this pipeline substantially. Wynn Gillis in the lab, Caleb Weinreb is working on the social stuff, and Diego and Scott on the hierarchical stuff, and then everyone else in the Datta Lab. And special shout out to John for taking this photo. I think that was you, right? Yeah, and happy to take questions. Yeah.
How adaptable is this to like a side view camera? Because I have some data that is from just the standard feed cage, and so they're doing behaviors in there. It's not as big as the area that you have, but is there anything in the internals of MoSeq that demand a top-down view?
Yeah, so the question is whether or not we could adapt the framework to work with a side view. So the current framework works using depth images. But there is work in the lab now to try and adapt the ARHMM to work with point tracking techniques. So there are some new behavioral tracking techniques now that use deep networks to track key points on animals, like deep lab cut. Oops. Deep lab cut is an example, and leap is another example. And in that case, I would expect functionality in the future for you to be able to use the modeling portion of this pipeline to pull out discrete states from key point tracking. Yeah, but you could theoretically record from the side with the depth camera and use this as well. Yeah.
Can you say a word about how effective this is in the context of drug screening? As in, can you see from the behavior the differences between drugs or modes of action or dose or things like that?
Yeah, so the question is, how well does this work on analyzing behavior to drugs? I didn't show any of this data, but there's been substantial work in the lab that is using this as a tool to basically identify what drug you have given a mouse. And so the answer is that yeah, you can predict with high probability not only what class of drug you've given a mouse, so stimulant or depressant or benzodiazepine, but also what dose you've given it as well. And that's work from the lab that should be published quite soon, I would say. So stay tuned for that. That's work from Alex Welshko.
I mean, a really related question is screening genetic models. How sensitive can you get, with this?
Yeah.
Just for understanding, motion behaviors for different models.
Yeah, it's quite sensitive. So in the original paper where the technique was published, we were investigating this ROR1 beta gene that is known to elicit this stereotyped kind of waddling phenotype in a homozygous mutant. It was actually unknown whether or not there was a phenotype in the heterozygous mutant, but we were able to show with this technique that there is. So I would say that for the case of genetic perturbations, this is the right technique. Yeah. Yeah.
So I think I might have missed this when you first talked about, but is this classification only done using posture information, or do you include any of the other sort of high-end velocity information that?
Yeah, good question. The question was whether or not we're just modeling the postural information of the mouse, or whether we're including any of the scalar information as well. So the work I described uses just the posture information, but we have used the additional scalar information, and it doesn't actually afford you much from a modeling perspective. Like the model performs better with just the postural data. Yeah.
Okay, let's thank our speaker again. Come back.
So I'm a joint postdoc in Bernardo Sabatini and Bob Datta's lab. And I'm going to tell you a little bit about what we've been up to for the past few years and where I think this work is headed. And if you guys aren't aware of what this painting is, so this is from Audubon. And I just usually like to show little slides from Audubon as the title slide for a talk, just because I think he really brings nature alive. These mice seem to have inner lives that sometimes I don't really see in our depth videos, so sometimes it's kind of nice to peer into their souls through these paintings. Okay, so anybody know what this is or what it's depicting? I'm asking mostly because it's before noon and we all need to wake up a little bit, but does anybody have any idea what this might be? Yeah?
It's a Minoan bull.
So you're the second person who knew exactly what it was. That's awesome. Yeah, so this is a fresco from about 1500 BC. It depicts a Greek practice known as bull leaping. This was a surprise to me when I first actually stumbled upon this. I guess it's a fairly well-known fresco just because it's so old and it depicts something fairly wild. So what it is depicting is actually somebody who walks up to a bull and decides to say hello to the bull by tugging on its horns. The bull obliges this hello by saying, okay, now I think I'm going to murder you. And the person then decides to summarily do a flip over the bull, land on the other side, and then do some sort of flourish for the audience, and maybe even a taunt to the bull. So the reason why I'm actually showing this is a fewfold. I think one, it shows something that's kind of wild and crazy, and maybe it'll wake you up a little bit. But B, I think it depicts something that's actually fairly profound about movement sequencing. So I think this has actually been something that we've been interested in as a people for thousands of years. You see many, many more paintings like this. And I think conceptually, what it shows in a single picture is actually the duality of movement sequencing. So you can see that it's depicting this sequence of bull leaping as actually 3 discrete categories of action. Each one of these categories actually has contained in it some fluid trajectory through space, right? So we would all agree that tugging on a bull's horns is different from flipping over it, and that, again, is different from this flourish at the end. But what each one of these things is actually a fluid sequence through time, and the tug on the horns is actually some smooth trajectory through space. Same with this acrobatics in the middle, and the flourish at the end. And so this is something I've been interested in for a long time. As Liz mentioned, as a graduate student, I originally worked in zebra finches. And this particular question, I think, has really been rattling around in my head for almost a decade now. And recently, as a postdoc, I've been asking these same questions in the mammal, specifically in the mouse. So this is a side view of a cartoon mouse's brain. And basically, all you need to know is what's highlighted in the middle. So for the past few years, I've been focused on this collection of nuclei known as the basal ganglia. So basal as in deep in the brain, ganglia as in these are nuclei of cells. They're not laminated like cortex. And so I kind of joke, and this picture maybe highlights this idea that cortex is kind of a helmet for all the interesting stuff in the middle. It's not actually true. Cortex does plenty. But for today, I'm going to talk exclusively about work in the basal ganglia. And it's a circuit that receives information from cortex. And then it sends that information through a collection of hops. And those hops actually get split into two subsystems. And this is maybe one of the most prominent features of the basal ganglia, which is there are two subsystems that can be defined anatomically, molecularly, and genetically, the so-called indirect and direct pathways. So the indirect shown here, the direct pathway shown here. The reason why they're called indirect and direct, the indirect pathway has a couple of hops before it reaches this output structure, the substantia nigra reticulata, and the direct pathway, which has a little bit of a shortcut, so it just has one hop to the substantia nigra reticulata. So for decades, we've thought that these two pathways have basically subserved two different things in the context of movements and movement sequencing. So we've commonly thought of the indirect pathway as one which suppresses or inhibits movement, and the direct pathway as one that maybe promotes movement. And so I think these two pathways, being such a prominent feature in a brain circuit that we know is critical for movement sequencing, there are still some outstanding questions about what this circuit is even doing. And so one big question is, how do these two pathways actually interact with each other? Right? So I just told you that maybe one promotes movement, maybe one suppresses movement. And I think one reason why we really believe this in the rodent is that there's been some really, really beautiful work from Anatol Kreitzer's lab with Alexei Kravitz, where they put a light-gated ion channel in direct pathway shown here as D1, since these neurons express the DRD1 receptor, or indirect pathway neurons shown here as D2 because they express the DRD2 receptor. So what they did was they dropped a mouse in a tiny little bowl and then stimulated the direct pathway or the indirect pathway. And these little dots and lines just show where the mouse was in the dish. If you stimulate on the direct pathway, the mouse kind of moves around a lot. And if you stimulate the indirect pathway, the mouse basically does a little spiral and then freezes. And this is a very dramatic result. It was, in fact, one of the first big results showing that optogenetics work well in vivo. And you can see in the summary plots in the bottom that these are some of the bigger effects you'll really see in behavioral neuroscience. So I think this gave us the idea that these two pathways are behaving antagonistically, right? So maybe one pathway really is pushing movement and the other is sort of pulling at movement. But then, a little bit later, through recording bulk fluorescence from these pathways, so here what this group has done is put a genetically encoded calcium indicator in either direct pathway neurons or indirect pathway neurons. If you record from hundreds of them at once, and you align that activity to, say, the turn before a mouse presses a lever, you see something that maybe seems naively paradoxical, right? So if you look, let's get a pointer here. So if you look right at the onset of the movement, you get a little burst of activity in both of these pathways. So if one's promoting movement and one's suppressing movement, how can this be true, right? This suggests something a little bit different, that maybe these two subsystems are really behaving synergistically, right? So now we kind of have two ways of thinking about how these two pathways interact. But I think there's been a parallel and I think equally important question, which is if we look in the basal ganglia, specifically in the big input region, the striatum that I showed you in the diagram before, what is actually the granularity with which these neurons represent behavior? So we know these neurons do represent behavior, but if you look at different experiments, you basically see things like this. So for example, in the primate literature especially, you see an argument that these neurons are coding for things like the flexion and extension of your wrist, right? So this suggests that maybe these neurons actually represent the kinematics of movement, right? So this is something like what you might call a continuous code for behavior. So maybe these neurons are coding for joint angles,
extension, flexion of the wrist, maybe even movements of your muscles. But then in the rodent literature, you see something that's actually quite different. So this is an example, really nice example from Anne Grable's lab. And what they did was lowered electrodes into the rat basal ganglia, recorded from neurons over the course of about a week while a rat learned to run in a T maze. So it's not really a maze, right? The rat just has to make one turn. So this is just a top-down view of this maze. Blocks indicate when neurons fire, and rows indicate the firing from the same group of neurons across time. So the first time you drop a rat into a T maze, neurons in the basal ganglia seem to really, really like when you turn in the middle. By the time you get to, let's say, day 9, these neurons seem to represent the beginning and end of the task. So this is something kind of different from this kinematic idea. If these neurons are representing kinematics, it makes sense that they would fire near the turn. But now they're actually firing at the beginning and end of the task. So this seems like something a little more plastic and a little more abstract. So I'm going to call this an argument that maybe the basal ganglia actually represents something abstract and discrete about behavior, say the beginning of a task and the end of a task when you get reward. So what I'm going to talk about in the first part of this talk is our efforts to really address these two big questions. How do these subsystems in the basal ganglia interact? And what is the level at which they represent behavior? Does that make sense so far? We're all on board? Good. All right, so how do we actually do this? So again, I'm showing you a cartoon side view of a mouse's brain, and we came up with a genetic strategy for expressing green and newly developed red calcium indicators. And we basically put them into viruses that allowed us to express them in the same mouse, but in separate cell populations. So this was new, and this was due to some technology that was developed by R.P. Saunders in Bernardo Sabatini's lab. And so here, we can actually visualize the activity of both the direct and indirect pathways at the same time in the same animal. Just to show you that this works, here's actually the expression profile in one mouse that's been given an injection of these two viruses. So you can see in red, it's a little dim here, but basically what you should be seeing, maybe in your mind's eye, there's a beautiful axon bundle coursing from the striatum, the main input region of the basal ganglia, to the SNR, which I told you is the output of the basal ganglia. Then in the indirect pathway, we've got a green calcium indicator, where this axon bundle is now going to another structure, the globus pallidus. Okay, so we can label both populations in the same mouse. We can pair this with some simple tricks that allowed us to collect green and red fluorescence with minimal crosstalk. This basically uses the same technology your radio uses. So it's really just a fancy form of frequency modulation that also uses phase information. That's what's shown on the left. On the right, shown basically as a circle, is MoSeq. So again, we're dropping a mouse in a bucket while we're recording both green and red fluorescence from the basal ganglia. And we're going to submit at least the behavioral data to an ARHMM. There are a few extra letters in here. You don't necessarily need to worry about them, other than we are using Bayesian inference to fit this ARHMM. And there's an S in front of the HMM. So this is actually the kappa parameter that Ralph referred to before. So S stands for sticky. If you've ever fit a standard HMM, they tend to switch like crazy. And so the S in there is actually to encourage the model to not switch as much. So beneath that, I think you guys are familiar with this type of ethogram-like picture. So each color here just indicates a different discrete state as identified by the model in the 3D video. And then here are some just lovely depictions of what these behaviors actually look like if you crack open those boxes.
So this autoaggressive hierarchical Dirichlet process.
You got it.
Sticky hidden Markov model.
Yeah.
Great. And so just to put a point on it, a state here, we learned in Ralph's talk that you're taking pixel data down to 10 dimensions using principal components, right? So, so we have a sort of mouse's pose represented as a point in 10-dimensional Euclidean space. And that point is moving around in 10-dimensional space in time.
That's right.
A state of this HMM is roughly a 10 by 10 matrix that describes an update rule for, given where the point is now, where will it be next? It's sort of a dynamical system on R10. and you're trying to find a finite number of these dynamical systems that do a good job of predicting the observed data given the structure of this Bayesian model by switching between them?
Yes. So I think your question, which was a beautiful description of the ARHMM, so just to give you a little bit of an idea of the internals of this model, so instead of a Gaussian emission, which you typically see in HMMs, this is a vector autoregressive process. So what that means is that every state in the model can be described by a 10 by 10 by number of lags in the AR process. In this case, that's 3. So that's 300 parameters plus another 10 for a bias term, plus noise. So each one of these states actually has a fair number of parameters that basically describes movement in this 10-dimensional PC space. Okay, so each state can be thought of as a basically linear dynamical system, right? So we're really just using inner products over this matrix and the principal components to describe how they evolve over time, right? So that's really the power of this model. So something that we didn't mention before is, the alternative to this would actually be to, say, compute a spectrogram of the principal components. So there's another way to capture information over time, and to do so over multiple frequency bands. It's a technology we've had for almost a century now, which is the short-time Fourier transform, right? So this model is actually directly modeling the raw time series rather than passing it through an intermediate transform like the STFT, which you could also do here, but requires another set of engineering design decisions. Yeah. Okay, so what does this actually look like in practice? So we're collecting green and red fluorescence from the basal ganglia, we're collecting 3D video, and then we're modeling that video with the AR HDP SHMM. Okay, so I'm gonna play the video, and this is probably a way of showing things that you're used to by this point. So on the left, we've got a centered and oriented mouse. This is what goes through the modeling. So this is just to focus on the pose dynamics of the mouse. And here we're actually showing, the details don't matter about this. These are random projections of the frames. The reason why one might use random projections here are actually to scramble the pixels so that the thing your eye focuses on are these little striations here. So you can actually see the structure, whatever it is, seems to change every 200 to 300 milliseconds. And this is one of the key insights I think Alex had in his first paper, which is mouse behavior looks pretty effing modular, right? I mean, this structure basically seems to completely remap every 200 to 400 milliseconds. It's a really striking feature of the data. So beneath that, these are the discrete states of the AR HDP SHMM, or ARHMM, its nickname. And each color now represents a different state identified by the model. Beneath that, activity of the direct and indirect pathways, right? So this is the new thing that we're adding into the mix. In red, we've got the direct pathway, and green, the indirect pathway. And so I told you before, people had seen that these two pathways seem to be active at the same time during movement, and that's not what you see here. What you see is that sometimes the pathways are synchronized, sometimes they're desynchronized, sometimes they're anti-correlated. So what's going on, right? What's happening here seems to be a little more complicated and subtle, I think, than what had previously been seen, by virtue of not being able to look at both pathways at the same time and not having this type of resolution over the animal's behavior. So the first thing you can do, which is what we all do, is take an average. So here what I've done is looked at every single transition that identified by the model. So every time you go from one discrete state to another discrete state, so a run to a jump to a turn, whatever it is. So if you look at every transition, that's shown here at time 0, there's a little blip in the direct and indirect pathways, and it's a significant blip. There's also a negative going blip, which is something we're still thinking about. But this positive blip is something that I showed you from bulk fluorescence imaging before, which is both pathways on average seem to be active whenever the animal changes what it's doing. So this is what people had seen previously. But it also tells us something already pretty interesting, which is the ARHMM does isolate a particular timescale of behavior, right? This didn't have to be true. It could have been that these neurons actually had, let's say, a 1-minute clock over the animal state. But the clock actually seems to be pretty matched, very well matched actually, to the clock that's found by the ARHMM. So, turns out, and this I guess was a nice stroke of luck, this area relevant for movement sequencing seems to have the same clock as identified by the model in 3D depth videos. Now, of course, the model identifies different states. So typically, the HDP, so the hierarchical Dirichlet process, will identify between 30 to 50 states, depending on what type of data you're looking at. But this is the number we typically see, especially in C57s. And so here, what I'm plotting is actually the average direct pathway and indirect pathway fluorescence for each of the 40 states identified by the model. And so I've just color-coded those states arbitrarily from red to blue, just so you can actually track different states across these 3 plots. This is now an average across all of our data. And the first thing that probably jumps out at you is that these 2 pathways are pretty correlated, right? This is something people had seen before. But what they hadn't seen was that each one of these points occupies a unique part of this scatter plot. So what this tells us is actually the specific balance between these 2 pathways tells you something about what the animal's doing. And also, there is a little bit of off-diagonal structure here too, so the correlation isn't perfect. Question?
Yeah, sorry, I may have missed this, but how are you accounting for the fact that JR camp and G camp 6 have very different delta F over Fs?
So the question is, these 2 calcium indicators that we're using that I didn't describe very well have different kinetics. So we tried to match the kinetics as best as we could. We also did basically the inverse experiment where we put red in the other population and green in the other population and saw the same thing. The other thing that we did was we actually went in and did electrophysiology while doing these bulk fluorescence measurements. And we basically found that the timescales, yes, they're different. They're not different by that much in this case. The other thing you can do, which is kind of a signal processing hack, is actually just take the derivatives of these signals. So JR camp and G camp, they have pretty different off kinetics. So what that means is when the neuron's not firing, after it is fired, the fluorescence takes some time to relax back to baseline. If you just take the first derivative of that signal, you get rid of the off. And it turns out the on kinetics are actually very similar. And basically, you can recapitulate these results just looking at the derivative of the signal. Does that make sense? Yes. Cool. Any other questions just about the techniques? OK. Or the data, whatever. Right. So we have a unique point in this scatter plot for each one of these 40 behaviors. And what's interesting is you get this structure in the average, but you also see this structure in individual animals. So this also didn't have to be true. So it turns out that the structure that we see in the average is actually conserved from animal to animal. So if you tell me the balance between these 2 pathways, at least to rough approximation, I can tell you what that animal's doing. Now again, this is skipping over some details, right? So I'm just telling you these 40 behaviors have different balances of activity, but we know that some behaviors are more similar to some behaviors than others, right? So 2 jumps should be more similar to each other than a rear and a walk, for example. And so we spent a little time figuring out how to basically probe the guts of the model to tell us whether 2 behaviors are more or less similar to each other. And so in brief, what we did, because these models have a set of autoregressive states encapsulating each behavior, we basically just played out those autoregressive dynamics. So we basically asked the model, why don't you play out your ideal jump, your ideal run, your ideal turn? And then we compared those to each other. So basically, this just denoises the data for us and allows us to build a nice distance matrix between each of the 40 behaviors. If you have a distance matrix, you can make a dendrogram, and that's what's shown here. And so our collaborator, Wayne Gillis, very nicely went through this dendrogram, every leaf of the dendrogram, and identified categories that you can easily isolate. So things like rears, walks, pauses, scrunches, scrunch and left turn, and so on. And what's cool is that you can grab 2 examples from 2 leaves of the dendrogram, look at the average D1 or direct pathway and indirect pathway activity patterns. And if you grab 2 points from within a category, they're more similar to each other than if you grab 2 points across categories. So what this tells us, at least a first approximation, that there's actually some systematic structure in the balance of direct and indirect pathway activity. So it's not just that they're different for every behavior, it's actually that they are different, and the ways in which they are different tells you what behavior the animal is performing. So we can quantify this a little more systematically. Basically here, what I'm taking are pairwise distances over the neural activity patterns aligned to the onset of each behavior. So that's what's shown here. And I'm also comparing that with the behavioral distance between any 2 behaviors. And if you look at the correlation between these 2 things using just the direct pathway, you get a kind of modest, crummy correlation. Same with the indirect pathway. But if you look at both, that's where you get a fairly strong correlation. So this is a highly filtered signal. So we're doing photometry, which is a way of collecting bulk fluorescence from hundreds of neurons. And still, we're actually able to resolve the similarity over behaviors, and that similarity is actually fairly precise, right? This is similarity in 3D videos picked up by our ARHMM. So this was actually pretty surprising to us. And this also tells us that you need information from both pathways to figure out what the animal's doing. You can't just look at the direct pathway and you can't just look at the indirect pathway. You really need information from both, which means they carry different kinds of information, right? So now you might say, okay, this is all wonderful. You found a bunch of beautiful structure, but maybe it's different. As soon as you change anything about what the animal's doing, maybe it just gets completely rewired. So we did kind of the simplest version of testing this, which is to take an animal, drop in a Petri dish on day 1. So this is just a position density in the bucket, and this is where the Petri dish is. Mice seem to like new things in the bucket. There aren't many features in it, so you put anything in there, it's like enriching the environment. These are 6 syllables we just picked out of a hat, and their average activity patterns in the direct and indirect pathways aligned to syllable onset. We can now put TMT in that dish, and what we see is for the same 6 behaviors, we get very similar activity patterns. And of course, we quantify this, and they are very, very similar. So it looks like even though we're dropping in a component of fox urine, which is what TMT is, and scaring the crap out of these animals, so they're in a very different state, it looks like, at least in the basal ganglia, the activity patterns representing behavior are basically frozen. But, a slight wrinkle to this, which is actually a super interesting wrinkle. So they're invariant to this kind of valence, right? So they're scared, but they're performing the same behaviors, maybe in a different order, and the activity patterns look similar. But then we can actually look at the sequence structure. So what do I mean by that? Well, we can look at all triplets of behavior, so 3 grams, align activity to the middle of those 3 grams, and now actually separate those averages by whether they're embedded in a high or low probability sequence. So what do I mean by that? Well, if the incoming transition probabilities are high and the outgoing transition probabilities are high, what does activity look like in the middle? If the incoming and outgoing are low, what does it look like? So what's happening in the middle is exactly the same. The only thing that's different is what kind of sequence it occurs in. And so what we found is that in both the direct and indirect pathways, low probability sequences have higher activity in the middle than high probability sequences. And so this seems very weird. They're doing the same thing. What's not weird about it is people had observed this type of phenomenon before. So what do I mean? So groups have basically found that if an animal performs a sequence of behaviors all the time, it basically gets chunked. So the basal ganglia, in some respects, is representing high probability sequences as one thing, but there's still a little blip in the middle, right? So it's basically carrying information both about the transitions between individual behaviors and the sequence statistics that they're embedded in, which is actually very interesting. So this is something we're obviously following up on, but for now, this is kind of where we are with the sequence structure. So this has all been bulk fluorescence measurements from hundreds of neurons. It's been pretty cool that we've been able to find this structure. But as Liz mentioned, you can use Schnitzer scopes, which are just miniaturized epifluorescence microscopes. Basically, they've used cell phone parts to make microscopes small enough for a mouse to carry, so on the order of 2 to 3 grams. It's actually a fairly amazing technology. And so what you can do is ask a mouse to just carry 3 grams on its head and carry a giant cable around with it. And then we can actually image the activity of individual neurons in the basal ganglia while still quantifying their behaviors exhibited in these 3D videos. So we went through some pains actually to computationally handle all the extra gear on their head. It's something you can do it pretty well. And that's actually why we use probabilistic PCA. So this was kind of a small detail that Ralph mentioned before. So what you can do is basically have a very, very simple model of what a mouse is, in this case a 3D Gaussian, and basically say from frame to frame, there's a blob moving around. And you can use the likelihoods from that 3D Gaussian to tell you what's mouse and what's not. And so what's not a mouse is basically missing data. And so then we can basically have PCA impute those missing values using an approximation of what a mouse looks like. And it turns out you run that iteratively, for 20 or 30 iterations, it works quite well. So we do that, we do our ARHMM, we get discrete states, and then we image the activity of a couple hundred single neurons in the basal ganglia. So now let's look at the single cell correlates of this structure. So first, if we just take a pseudo-color plot of one such experiment, so here we're actually looking at about 300 to 400 neurons over time aligned to the discrete states of the ARHMM, or the behavioral syllables. And what's kind of interesting here is you can see what was going on in our bulk measurements, which is these neurons are fairly sparse, so they kind of clock at around 0.1, 0.01 hertz, right? So they're not even firing once a second. But there are points where many neurons in the basal ganglia are firing. And it turns out more often than not that these periods where they're firing, where they're synchronous, are periods of behavioral transition. And this is what we had seen in the photometry data, which is there's a little blip in both the direct and indirect pathways when they transition from any behavior to any other behavior. So it turns out what's underlying that signal is the synchronization of this network in the basal ganglia. So what was underlying all that systematic structure I told you about? So similar behaviors have similar activity patterns, and dissimilar behaviors have dissimilar activity patterns. Well, it looks like what's going on is this. So here, I'm just going to walk you through this slightly complicated plot. What I'm doing is taking all the neurons we recorded. So this is about 1,200 neurons in about 10 different animals. And I basically just mashed them all together into a pseudopopulation and then sorted their activity aligned to the onset of a behavior. So everything in this column is aligned to a scrunch. Everything in this column is aligned to a dart and turn left. Everything in this column is aligned to a scrunch left. And so now I've sorted these neurons by their activity around the scrunch. Okay, so I've just sorted from positive peak first to last, negative peak last to first. So this just gives you a full profile of what all of the neurons and all of the mice did for a particular behavior. So if I align to a scrunch and I maintain that alignment when the animal darts and turns left, you basically get a little shadow of this structure. And it's actually fairly prominent here. So you can see in the scrunch turn left, you get these nice little swoops, which should be there because that's basically what should happen when we align the data. And you can see basically there's a little shadow of that stripe in the dart and turn left. So for similar behaviors, basically a subset of the neurons are participating. So it looks like what's going on is we've got a bunch of neurons that very clearly represent the scrunch and turn left, and a fraction of them are also firing during this dart and turn left. So it looks like there's this relational code where populations of neurons are probabilistically firing during certain behaviors, and they have a higher likelihood of firing during other similar behaviors. Does that make sense? Maybe? Good. So everything I've told you so far is correlative, right? So we've gone and looked at activity patterns in the basal ganglia. They seem aligned to these behaviors. You might, if you follow the literature in systems neuroscience these days, it looks like almost every brain area is correlated with behavior. And I guess that's what the brain does, so maybe this isn't a surprise. So we needed to do something else to actually demonstrate that this brain area does something relevant for what we care about. So we did probably the simplest thing you can imagine. We injected NMDA into the region that we were recording from. What NMDA does is basically excite these neurons until they explode. So basically, we just killed off a fairly large fraction of the mouse's brain and then watched it behave. Now, I'm going to play you basically 10x sped up footage from a sham and lesion mouse. And when we looked at these videos ourselves, we kind of had an oh crap moment because we didn't really see anything that stood out in these videos. So, maybe you can find something that's different about this lesion. I told you it's a lesion, so you're already looking for it. But if you just watch them, they still run, they still rear, they still investigate the bucket. Maybe it's just that the bucket is impoverished, right? But it looks like they kind of do all the same things. Even though we've obliterated, a substantial fraction of the mouse's basal ganglia, which I told you is essential for movement. So what's going on? So it turns out that basically all of the effects were in the sequence structure of the mouse's behavior. So here, what I've done is just taken all 40 behaviors, arranged them into a ring, and then I draw lines in between them, and the width of those lines tells you how likely that bigram is. So how likely it is that one behavior follows another behavior. So it's really likely that you go from a run into a rear, for example. So this is what the structure looks like in a sham mouse. Turns out there's a substantial amount of structure. There is very clear first-order Markov structure, maybe even second order. But if you look in the lesion, basically everything's different. So if you compare these two, they're drastically different. And what seems to be going on, so here in blue, that indicates that bigram probability went down. Red indicates it went up. A lot of the weak links are going up, and a lot of the strong links are going down. And so the net effect of that is actually to make their behavior more entropic and more random. So mice normally have some structure to how they sequence their behavior, and a lot of that structure goes away when you lesion the striatum. So it's a very specific effect, but it's actually one you would have anticipated if you'd looked at Kent Berridge's work, something that Ralph also mentioned from the 80s and 90s, where they actually saw this type of effect in the grooming syntax of rodents. And we see basically the same thing in all of their behavior in the open field. Yeah?
Just to make sure that I understand. So if I were looking at the fingerprinting behavior, so the plot of frequency of a syllable, would the two plots look the same?
No. So that's also different. What's not different is the execution of the behavior. So that's what's interesting. So if I actually look at a rear in the sham and lesion mouse, it's exactly the same rear. What's different is how often they do it. So that is different, and this is more different. Does that make sense? Yeah. So basically, interestingly enough, this was kind of like a perfect use case of this platform in neuroscience, where the effect was not necessarily in the execution of behavior, which we could validate by going through our 3D videos. It was really in how that behavior is sequenced. So just to conclude from this part of the talk, I told you the direct and indirect pathways are active on average. It transitions between syllables, and the ratio specifies the identity of a given behavior. I've told you using dual color bulk fluorescence photometry that there's a systematic relational map of behavioral syllables, and the direct and indirect pathways contain parallel and complementary codes for behavior. Using cellular resolution calcium imaging, I've shown you the single cell correlates of the structure we observed in the bulk measurements. And lesions really demonstrate the basal ganglia's role in action selection. Right? So it seems to really, specifically be deciding what action to perform on a moment-to-moment basis. So I'm going to transition into some unpublished work that's kind of been, something I'd been dreaming about for a long time. So in the songbird community, they have these beautiful closed-loop tools, so they can isolate the particular note a bird is singing with 100 microsecond precision using FPGAs. And I thought, man, it would be super cool if we could do something like this in a mouse. And the reason why this is interesting in the context of this particular circuit is, I've told you a lot about these red and green dots, direct and indirect pathway neurons in the basal ganglia. So something that I kind of swept under the rug was that there's this huge projection from the midbrain that's dopaminergic. So the substantia nigra compacta and the ventral tegmental area send these very dense dopamine projections to the basal ganglia. And you've probably heard of dopamine in many contexts. And what people think it's doing here is actually implementing something called reinforcement learning. So basically, how does a mouse learn to do some movements at some times? How does it learn to shape those movements? So this really brings learning into the picture. And so we thought, OK, how can we do something closed loop here that tells us something about how the system really works in a freely behaving animal? And we were inspired by an X-ray of a dinosaur, which is actually a rat. So when you look at this is actually kind of crazy. This is an X-ray through a rat's skull. And so I was looking at this a couple of days ago, and I was like, what is that? So those are the incisors of a rat, which is why a rat bite really hurts. But what's really relevant here is actually this electrode array, which is implanted up here. So this is actually a picture from an experiment that Olds and Milner had done mid-20th century, which had basically spawned a field called intracranial self-stimulation. So what is that? So they had the insight that you can implant electrodes anywhere in the brain, but here you get really, really dramatic effects if you put them near the basal ganglia or the midbrain. What you can do is put the electrodes in the rat's brain and basically give rats control over those electrodes. So every time the rat presses on this lever, it gets a little current through those electrodes. And so I think in this particular picture, the argument is that this rat is feeling pleasure. I don't really know how to interpret this, but there you go. I guess it's pleasurable. And actually, if you look at videos of these experiments, amazingly, if you electrify this floor, they will walk over fire, basically, to get to that lever press. They love that lever, especially if those electrodes are in the basal ganglia or in the midbrain. So dopamine is basically a very addicting and reinforcing stimulus to these animals if you give them direct control over it. And so the thought that we had was, OK, why don't we take MoSeq and basically give rodents the ability to give themselves dopamine if they perform the right syllable. So how do we actually do this? So you guys know what MoSeq is. That's a diagram of MoSeq. Who cares? What we can do with that, though, is get some labels if we MoSeq some baseline data, and we can look at features that we can easily compute in real time. So things like velocity, height, width, and length. You can come up with a bank of 20 reasonable features for computing here. So these you can compute in a real-time system, basically in under 10 milliseconds. It's not a big deal. But the thing that you need to learn is some mapping between those features and the syllables identified by MoSeq. And so we did, I hesitate to call this a simple thing, but I guess it's simple these days, which is fit a deep network between those features and the syllables. So the structure of that network is shown here. The details actually don't seem to matter all that much. But in short, what we did was we basically created a set of 1D convolutional layers, fed those through an elaborated multi-layer perceptron, and that learns a nice mapping between these video features and those syllables, and it can run in real time. So here's just the round-trip latency of this system. It's nowhere near where the Songbird folks are, which is all under a millisecond. So here, the round trip is basically the time from a physical event in the world to a physical event that might be, say, a pulse that triggers a laser. And so the round trip is about 50 milliseconds here, which is good enough. Since, as we told you, the clock on mouse behavior in this system at least seems to be roughly 300 to 600 milliseconds. So it seems like we can recognize behaviors and give them a little dopamine before that behavior ends. And here's just a confusion matrix demonstrating that the system is also fairly accurate. This is getting better all the time, and we're also thinking of other ways to actually implement this. This is kind of the duct tape deep network solution that we're using now. And I guess the proof is in the pudding, right? So you'll see basically how well it works in a few slides. Okay, so we've got a parts list of behavior, and we're basically going to try and isolate 3 syllables and give the mice a little dopamine whenever they perform them. So I mentioned that there are these 2 dopamine projections from the midbrain. We've done experiments in both, but I'm only going to describe experiments in one of those subsystems. So what we did was we injected a light-gated ion channel into the VTA and then put optical fibers above their projection to the ventral striatum, which is a part of the basal ganglia that receives a very dense dopamine projection. So every time we pulse blue light, we're basically triggering the release of dopamine in this specific brain area. And so the protocol, which is basically something we picked out of a hat, so there are probably ways to improve this. We record some baseline data in one session, and then over 3 separate days for 30 minutes each, we're going to give the mice stimulation whenever they perform a particular behavior, and then we're going to switch the behavior on subsequent weeks. So what actually happens? Can they do it? It turns out, yes, the answer is they can do it, and they can do it very well. So here, what I'm showing is actually how much they enrich just that one target syllable over baseline in the stimulation condition. Okay, so I'm taking the probability that they express just that one behavior and dividing it by their baseline expression of that syllable. And basically, they can push it up by almost 30% in some cases. And they can do so without actually pushing up any similar behaviors or dissimilar behaviors. So we can get a lot of resolution over this and basically ask, okay, if I look at the 3D pose trajectory and I look at, say, another rear, another walk, another scrunch, turns out they're not pushing up those other behaviors. So the mice aren't confused by this protocol. They know that syllable 37 is syllable 37. And so really, this also validates to a certain extent, I think, the segmentation sped out by our algorithm.
So how does that square with your comment before that there's overlap in the actual neurons that are driving similar behaviors?
That's a great question. So the question is, I told you all this stuff about relational maps, and the basal ganglia seems to have some access to whether behaviors are more or less similar to each other. Right? So I think both of these things can be true. So the neurons can represent similarity, but that doesn't necessarily mean that reinforcement has to basically bleed through from one behavior to the next. So you can imagine, let's say I reinforce a behavior 30 times, and basically what's going to be reinforced are the neurons that fire all 30 times. And so the same, let's say the same behaviors also fire probabilistically for other syllables. If you take enough of an average, over 20 to 30 trials, perhaps that's how it's solving this problem. And that's actually my intuition about this circuit, is you may want the relational map, but you may also want to be able to reinforce individual behaviors. And the way you could do that is by basically doing some trial averaging. Right? So if you don't just learn 100% after the first stimulation, which is not what happens, then you might get a lot of that kind of crosstalk. right? Right, so we don't see this type of learning if we use EYFP instead of channel rhodopsin, which is good news. They're not just learning to blink a blue light. And this learning seems to get better across 3 days. So how quickly can they learn within a session? So something I didn't really mention is that typically when you train a mouse to perform a behavior, it's painstaking work that can take days, weeks, sometimes months, depending on the complexity of the behavior. This one they seem to get within about 5 minutes. So we drop them in the bucket. The probability of the target basically goes up very rapidly. So it seems like there's a bit of an aha moment where they get 10 to 15 stimulations. The probability that target gets reinforced, and then that learning seems to maybe kind of sort of get refined over the course of the next 15 to 20 minutes. So they get a little dopamine, they want to get more dopamine, they figure out which syllable to perform in order to continue receiving dopamine, and all's good. To give you an idea of what the dynamics of this learning look like, here in this mildly complicated plot, I'm showing the probability of expressing the target syllable in red, and then I'm showing all of the other syllables color-coded by how similar they are, from white to deep blue. And basically what you can see here is red is kind of small in area, and then it expands. So this is maybe the aha moment. And then over the course of learning, some of these other neighboring syllables seem to go up and down. So now we're really interested in what are the fine timescale dynamics of how they're changing their behavior in response to this. And of course, the other second order question is, how does all this structure in the basal ganglia change while we do this type of experiment? I don't have that answer for you now, but really interested in it in the medium to long-term future. Now, something, of course, that I have to show you is that they're not just learning to do this by changing some other aspect of their behavior. So you might expect, ooh, you're pulsing dopamine into a bunch of neurons that represent movement. So maybe there's some acute effect of that stimulation. That's here. So in yellow is our stimulation. And then I'm just showing you velocity, height, width, and length. In our channel rhodopsin mice and our EYFP mice, we don't see any difference. So there doesn't seem to be an acute effect on the behavior. Maybe they're learning place preference. Maybe our system is just broken, and every time they go to the bottom part of the bucket, they get a little stimulation because of some goofy way we wired things up. We know you can stimulate these neurons to induce a place preference. That's not what's happening here. So it seems like they hang around the edges of the bucket. So somebody had a question before about whether it's a random walk. It seems like they, even after 3 days, like to spend a fair amount of their time near the edges of the bucket. And we don't see any overall changes in how they execute behavior. So as far as we can tell, there aren't any off-target effects. What about the global structure of behavior? Maybe just everything is getting remapped, and they're figuring out whatever it is to get another life-saving, life-affirming squirt of dopamine. And so here, what I'm showing is the average height and 3D velocity of each one of the 40 behaviors identified by the model. I've just assigned each behavior a color code, and that's preserved from the baseline through to stim day 3 for the 3 different behaviors we reinforced. The target is shown as a little white dot here. And basically what you can see is it's not as if all the behaviors are just collapsing into that one behavior. It's not as if they all of a sudden turn a jump into a walk. And you don't even see a lot of movement in the neighboring behaviors. So at least first pass, this seems to argue that these syllables are really discrete things, and what they're modulating is the probability that they express one of them. So that's all I've got for the reinforcement learning part, and I think with the last 2 minutes, I want to talk about what's been a little bit of a side project that's turned into a not so side project, which is to apply these time series models to mouse vocalizations. So there's been a lot of excitement, especially since this paper from Tim Holey, where they very beautifully described vocalizations in laboratory mice in the ultrasonic range. So that's above 60 kilohertz. So it turns out if you do really fast recordings, you can find these almost, if you squint your eyes, songbird-like squeaks that look like different notes. And so something that we thought might be interesting would be to take some of this data and actually apply some of the modeling techniques we use for 3D video to this data. So somebody asked, can you use the internals of MoSeq for different types of data? And the answer is, of course. Anything that can be represented as a time series can be subjected to this type of modeling. So what does this pipeline look like? It's provisionally called VOSeq, but I'm open to suggestions for what that name should be. Everything doesn't have to be a variation on MoSeq. So this is not our own data. This is data from Lisa Stauer's lab. And basically, the pipeline is a little bit different for what we do with MoSeq. And so one reason for that is that vocalizations are very easy to segment because animals take a breath, right? So the reason, and I think this is actually one motivation for why you would want to use an AR-HMM for mouse video data, which is they don't stop behaving. It's a dense time series. So there are no points where they stop and wait to do another behavior. It's they're stopping, they're going, they're turning. So you need a model like an AR-HMM to tell you where are the change points and how many different behaviors are there. But here they're taking breaths. So it's a very easy thing to segment. So we can take a bunch of vocalizations and detect power in the ultrasonic range. Then we can take all these vocalizations, basically crop them. So here I've just taken, looks like maybe 30 vocalizations, smash them end to end. So this is a spectrogram. If you're not used to looking at spectrograms, it's sort of like sheet music. So this is frequency, this is time, and the heat just tells you how much power there is in any particular time frequency bin. So this is what some mouse vocalizations look like. And here what I've done is actually time warp them. So this is another thing that's a little bit different from how MoSeq works. Because we know when the beginnings and ends of these vocalizations are, we can easily just time warp them so they're all the same length. So they're mostly continuous contours in time and frequency, so it's a very easy thing to warp. You can do dynamic time warping, other ways of doing a nonlinear warp, but in this case, just linear warping, i.e. interpolation, is really good enough. And so what that means now is that we have a 2D image that represents each vocalization. We can use standard matrix decomposition. So sort of like what we do with MoSeq, where everything gets crunched down with PCA. We can take a bunch of vocalizations, make them rows in a 2D matrix, and a time frequency pixel now is a column. And we can do PCA on this big matrix and have it tell us what are the important parts in the spectrogram for classifying mouse behavior. You can do other ways of matrix decomposition, and I'll show you what that looks like at the end. So this is what eigenvectors in PCA look like. So you can see that PCA is basically picking up sweeps, which is what you can pick up in the spectrograms, right? So mice basically just whistle and squeak a lot. There's not a lot of complex spectrotemporal structure. You can capture most of the information, both in time and frequency, with 17 to 18 principal components, which is actually pretty good because there are 500 to 1,000 pixels in each vocalization, depending on how you compute your spectrogram. And this is the output. So the underlying model that we actually use here is just a Gaussian HMM. These aren't time series anymore. These are little images that also have transition structure to them. So what's interesting is, and this is something I hadn't really known before, which is mouse vocalizations seem to do have some Markov structure, some first order transition structure to them. And that's useful when doing clustering. So each row here is a different cluster identified by our HDP, hierarchical Dirichlet process HMM. And you can see that all the vocalizations in a row look more or less similar to each other. So this looks like a pretty reasonable parcellation of the vocalizations by my eye, at least, based on what I'm used to seeing in songbirds, which have very categorically different vocalizations. These are definitely messier, but I think they are separable. And you can get roughly, I would say, 15 to 20 different categories of vocalizations from these mice, which is actually really cool. And we haven't really done much biology with this yet, but I'm actually just kind of excited to dig in and see what kind of structure there is in these vocalizations and potentially how the brain controls them. So I mentioned previously, there are other ways to project this data. I'm sure most of you are familiar with UMAP, so we UMAPed the data just to see how it would compare with principal component projections of the data. And of course, you get these weird looking, I spilled a little bit of oil on the shop floor kind of picture of the data. So this is UMAP projection of a few hundred spectrograms of mouse vocalizations color-coded by the HMM segmentation. So the HMM is using principal components, not the UMAP data. So it actually looks pretty coherent to me, which is cool. And then you can do something which isn't all that useful here, which is basically to show the transitions between each vocalization in that space. So anyway, this is just a visualization. There's no real biology here, but it's just something I've been working on that I think is interesting. So I'll stop there. There's a cast of characters to thank in both labs. So I'm joint between the Sabatini Lab, ostensibly interested in the basal ganglia, among other things, and the Datta Lab, which is interested in odors and behavior more generally. Of course, Alex and Matt need to get a big thanks because most of what I've talked about is used technology that they originally developed. Wynn Gillis is a graduate student in Bob's lab who I've worked very closely with for the published work and the unpublished work. Diego Alderondo is a rotation student who we're hoping will not be a rotation student at some point and will join the lab because he's awesome. Ralph, of course you know, has really been an integral part of all of these experiments and is a really amazing utility player in the lab. Emily, who I'm told is not related to Ralph, even though she has the same last name, is also a technician in the lab, has helped for pretty much every experiment I've described. Rob is an amazing undergrad who's been in the lab for a few months, who's basically been running the reinforcement stuff on his own. And we have an amazing lab manager, and of course I should thank Bob. In Bernardo's lab, there's a bunch of people who helped out both with the design of the vectors that we used for expressing the green and red calcium indicators, that's MoSeq. And these folks were really, really great in helping us collect lots of data with these miniaturized epifluorescence microscopes. Of course, I should thank Bernardo. All the vocalization data came from Lisa Stauer's lab, in particular a postdoc, Zheng Yi. And you've probably seen Scott's name a million times, and that's for a good reason, because he is awesome and he knows everything about these types of models. So he's just been a spectacular resource, and he's going to be starting his own lab in Stanford, I think, in the summer, the fall, sometime soon. And the folks that run the core facilities at HMS, we wouldn't be able to do most of what we do without them. And the building manager, Steve Sloboda, who basically helps with all the things that break. Funding to thank, and I try to come up with different ways of combining Bernardo and Bob's names. So I think I've got Bob Nardo or Sandardo Sabadatini. I'll also take suggestions for what to do here. So when you have joint advisors, you kind of fuse them in your head. And so I'm trying to sort of literalize that fusion. And one other thing, which is, this I think is the ultimate validation of MoSeq. So this was, I think, when I was preparing my first presentation and at like 2 in the morning. And one of the cats my wife and I had basically thought that there were mice in the monitor that needed to be relieved of being inside of the monitor so they could become dinner. So basically, this cat Amber spent the entirety of me putting together this presentation trying to capture these mice so she could consume them, which must mean that our representation is good enough to fool a cat, right? They have good visual systems, so maybe that says something. Anyway, thank you very much.